Hello everyone. Welcome back to Tamali's biology classes. Today we'll talk about a new topic known as signal transduction. This topic comes under the unit of cell communication and cell signaling. Let's start with the topic. Wait till the end because I have some questions ready for you, which you can go through for your exam preparation. Okay, so let's start. First of all, what is signal transduction? See, we know that our body also needs to respond to external stimuli, right? To the temperature changes outside or to the outside environment. So this communication with the outside environment, this communication with the stimuli that is received by our cell, that is called as cell signaling or cell communication. This communication we'll be studying in this part. So first we'll talk about what is cell signal transduction. Okay, so as I've told, our cell, they contain some receptors. Okay, so these receptors, what they do, they will detect some signaling molecules or in the form of like ligands. So what they will do, they will receive those. Okay, they will receive those and then they will pass the signal to the internal part of the cell. Okay, to the interior part of the cell. So this process by which the cells will receive and respond to signals from the environment is known as signal transduction. So this is very, very important because this communication has to occur inside the cells for the cell to grow, for its survival, for differentiation, and for other processes that occur in the cell. Fine. Now, what are the components? See, every process has some components. So in this part, what are the components? First of all, as I told you, there has to be a receptor. Okay, this receptor it may be a protein which will bind to the signaling molecule. As I told you, it's a ligand. Okay, so what happens? Ligand is present in the outside environment. So on the cell surface, there are some receptors which will detect them. Fine. After detecting them, this is passed on to the transducer. So what the transducer does, what it will do is it will convert the signal in the form by which the cell can understand. Okay, so there is a work of transducer, and after that there is an effector in the interior part of the cell. This effector will carry out the cellular response. Now let's come to the cell surface receptors. So these receptors, as I told you, they are embedded in the cell membrane, or you can say plasma membrane. Fine. So they will allow the cell to communicate with the outer environment with the help of ligands. Now let's talk about the structure of the cell surface receptor. So there are three domains of cell surface receptor: the extracellular domain, which contains the ligand binding site. Then we have the transmembrane domain, which will be present between the outer and the interior part. Then we have the intracellular domain, which will transmit the signal inside the cell from the external environment. Okay. Now there are three types of cell surface receptors. So what are they? One is ion channel receptors, one is G protein coupled receptors, and one is enzyme linked receptors. I have given a diagram on the right hand side. You can see out here. See, this is the ion channel link receptors. Okay, I'll show you. You can see here, the first one is the ion channel receptors. So what happens out here is some ions are detected, they come. Okay, so this channel will open as soon as the ligand comes and bind. Okay, with the ions, they will come and bind here. It will lead to opening of the channel. When the channel opens, there's a change in the membrane potential. Okay, after that again the channel will close when the the signal has been already transduced and it has already been sent into the interior part. In this form, in this part, we're talking about the ions. Okay, when this is already the ligand which is already going inside. So what will happen out here? The ions are being sent inside the cell, and then the channel will close because it happens because of a change in the membrane potential of the cell membrane. Okay, now this is these ion channel link receptors. Then the enzyme link receptors. Out here, one example is of tyrosine kinase. Okay, so here same thing will happen again. The ligand will bind out here, and tyrosine kinase will help in the further process out here. Then the next one is the G protein coupled receptors. We'll talk about all the three types in details in my future slides. Then we'll talk about the second messengers. See, in transduction pathway, what happens is that after this ligand, after this signal is taken in by the cell, it leads to there will be some messengers inside. Okay, there will be some receptors who will bind to them and who will activate the messengers. Okay, so these messengers, what will happen? They will come and they will help in the further processing of the signal. That means there are some downstream responses, downstream signals, which will be brought about because of this second messengers. Now, these second messengers or secondary messengers are of three types. It may be cyclic, okay, then it also may be you're in the form of lipids, and it also may be in the form of ions. Okay, now what is happening? First, we'll talk about the cyclic part. So, what are secondary messengers? So, they have the ability to bind to the protein targets and they will change the activity and thus lead to the downstream signals or responses. Some examples of secondary messengers out here are CAMP, CAMP, calcium ions. Then we also have I3P, or you can say inositol triphosphate, and DAG, also known as diacylglycerol. Okay, so this CAMP and CGMP, you can CAMP or CGMP, these two are coming under the cyclic nucleotides. Fine. So what does the CAMP do? CAMP will help in the activation of the protein kinase K. Okay, protein kinase A, PKA. Okay, and also a serine, which PK is actually a serine threonine kinase. Fine. And what does this CGMP do? CGMP will help in the vasodilation process, phototransition process, and neurotransmission process. Fine. And CAMP will help in the regulation of enzyme activities, ion channels, and gene transcription. We'll see the function in the future slides, how they are helping, how they're acting and all. Okay, just so here I've just shown you a simple diagram where I've shown the activation out here. You can see the activation part out here. See, in the activation part out here, you can see this inactive PKA. Okay, just a minute. Okay, you can see here the first, this PKA out here, this is an inactive PKA. So it has got regulatory subunits and it has got catalytic subunits. So when the ligand come and binds out here, it will lead to the catalytic subunits being active. Okay, when this CAMP will come and bind out here, in this case CAMP is coming up because ligand तो in the first step it has already come and bound. Okay, पहले में क्या होता है, ligand आके bind हो गया, receptors आके activate कर लिया secondary messengers को, and then when this CAMP, when it will come and bind, then what will happen? This catalytic subunits will get active. First, it's first in inactive format, the regulatory subunit. So first they'll come and bind regulatory subunit, और उसके बाद it will lead to the activation of the catalytic subunits, and इसके बाद बाकी जो downstream process होगा वो होगा. ठीक है? Now the other part, let's come to the second and third type of secondary messengers, the lipids and the ions. तो पहले हमने देख लिया कि CAMP and CGMP are the cyclic ones. In lipids we have the DAG and we also have the IP3. तो DAG and IP3 यहाँ पे मैंने दिया है in my notes that DAG and PIP2 are both lipid molecules, but DAG, PIP2 is not a secondary messenger, okay? But PIP2 क्या है? It's a precursor, okay? Phospholipase C, it will activate this PIP2. You can see in the flowchart that I have made out here for a better understanding. Here you can see phospholipase C will activate this PIP2, okay? And then this PIP2 will help in the breakdown. Will break down to form DAG and IP3. DAG will activate the protein kinase C, PKC, and IP3, okay? It will control the calcium levels in the cell. So IP3 कहाँ पे होता है? It will go to the cytoplasm, and from there, what will happen? It will go and bind to the receptor present in the endoplasmic reticulum. वहाँ जाके क्या होगा? As soon as this binding occurs, it will lead to release of the calcium Ca2+ ions. Okay, this Ca2+ will activate the calmodulin. This calmodulin will mediate inflammation, metabolism, cell death, immune response, and memory. These are all the downstream responses. Okay. Now coming to the DAG part. DAG will activate PKC, and then this will phosphorylate the proteins and lead to the signal transmission and also help in regulation of cell growth, ion channel functions, secretion, phagocytosis, then neurotransmission, gene expression, and metabolism. These are again, ये सारे क्या हैं? These are all the downstream responses which are occurring because of the activation of protein kinase C. Okay, so this is the process that is occurring. तो ये सब कब होता है? जब GPCR, that means G protein coupled receptors, जब वो activate हो जाते हैं, then what happens? It leads to the formation of IP3 and DAG from PIP2. Okay. Now coming to the ions part. अब ions में आ जाते हैं. What are the ions which are acting as secondary messengers? We have Ca2+, we have potassium, and we have magnesium ions. Fine. So यहाँ पे what do they do? They act as cofactors for enzymes and structural proteins. Okay, they will help in the transmission of nerve impulses. They will help in controlling muscle contraction. We know that Ca2+ ions वो क्या करते हैं? Ca2+ ions क्या करेंगे? They will help in the muscle contraction, neuronal transmission, okay, cell motility and cell growth. Fine. Now let's move on to the stages of signal transduction. हमने अभी देख लिया कि क्या क्या components है, उनके functions क्या क्या है. Now let's talk about the stages. Stages में क्या क्या है? 3 stage है. पहले मैंने ऑलरेडी बताया, मैं फिर से रिपीट करती हूँ. रिसेप्शन होगा, जहाँ पे जो रिसेप्टर्स है वो क्या करेंगे, दे विल एक्सेप्ट द सिग्नल मॉलिक्यूल एंड देन उससे क्या होगा, दे विल पास इट टू द ट्रांसड्यूसर. ट्रांसड्यूसर क्या करेंगे, उस सिग्नल को एक ऐसे फॉर्म में कन्वर्ट करेंगे विच इज अंडरस्टूड बाय द सेल एंड देन इट विल लीड टू द रिस्पॉन्स, द डाउनस्ट्रीम रिस्पॉन्सेस. ओके, तो ये है आपका स्टेजेस ऑफ सिग्नल ट्रांसडक्शन. नाउ व्हाट आर द टाइप्स ऑफ रिसेप्टर्स, वेरी वेरी इंपॉर्टेंट. देयर बहुत सारे हैं. तो here I've mentioned about 3, that is G protein coupled receptors. Then I've mentioned about receptor tyrosine kinases, RTKs भी बोलते हैं, and ion channel receptors. Okay, so G protein receptors क्या करते हैं, they will activate the G proteins and then RTK will phosphorylate. RTK क्या करते हैं, वो खुद को phosphorylate करते हैं, आसपास के proteins को भी वो phosphorylate करते हैं. Okay, and this will lead to the initiate the other signals or you can say the downstream processes. एंड आयन चैनल में मैंने ऑलरेडी बताया था, वो आयन चैनल को ओपन एंड क्लोज करते हैं. कैसे? बाय चेंजिंग द मेम्ब्रेन पोटेंशियल. तो यहाँ पे मैंने एक डायग्राम दिखाया है. तो डायग्राम में मैंने जस्ट यहाँ पे टाइप्स बताया है, तो आप देख सकते हैं, यू कैन सी आउट हियर. So this one is the first. Okay, here one more I have mentioned out here about intercellular receptors. This is also present. What happens out here? The communication is occurring inside the nucleus. This leads to gene transcription. याद रखिएगा. This leads to formation of DNA to mRNA. Okay, ये जो ट्रांसक्रिप्शन प्रोसेस है यहाँ पे होता है inside the nucleus. This is the intercellular receptors. Fine. Then we have the ligand gated ion channels. Okay. Then we have the G protein coupled receptors. जहाँ पे I've already mentioned about the secondary messengers. Already देखा आपने what are the second messengers and how they are helping in the cellular effects. Then we have the enzyme linked, where I've already told you about receptor tyrosine kinase. Okay, here it will lead to protein phosphorylation and gene transcription. So we will see about this in details. So first we'll talk about the G protein coupled receptor. So you can see the diagram out here itself. What is this? This is a class of membrane protein receptors. इसको 7 TM receptors भी बोलते हैं. TM means transmembrane. Okay. So what's happened? They have got 7 alpha helices out here. This G protein coupled receptors. और यहाँ पे देख सकते हैं बाहर में कितनी loops है? 1, 2, 3. 3 loops outside and there are 3 loops inside. आउटसाइड टर्मिनल है N टर्मिनल और इनसाइड में है C टर्मिनल. ओके, तो बाहर में क्या करते हैं, इट विल अगेन बाइंड टू द लिगैंड आउटसाइड और कैन से मॉलिक्यूल्स और एक्टिवेट करेंगे विच विल लीड टू द सिग्नल ट्रांसडक्शन पाथवेज. ओके, सो यहाँ पे ये है. नाउ क्या होगा, दिस विल, अच्छा यहाँ पे क्या क्या फंक्शन होता है, देखिए क्या क्या फंक्शन में हेल्प करते हैं लोग. दे हेल्प इन दी विजुअल कंट्रोल, किडनी फंक्शन, tumorigenesis, immune response and inflammation. Now first step, what happens out here is the first step तो होता है कि ligand binding होता है. Okay, these are the steps, but मैंने बस ये वाला अलग से रखा है, G protein activation. The first step is always the ligand attachment of ligand. Okay, first हमेशा ligand binding होगा. So after the ligand is bind, there is a change in the structure. तो क्या होता है कि conformational change होता है, receptor protein में conformational change हो जाता है. And then क्या होगा, G protein activation. This is the second step, G protein activation. So what happens out here, I'll tell you in detail because this is important. See out here, this is a heterotrimeric unit. Okay, देखो G alpha, G beta, G gamma. It's a heterotrimeric unit. So this G protein has got 3 subunits, alpha, beta, gamma. So before क्या होता है कि G alpha is attached with GDP and it's in a inactive state. As soon as ligand comes and bind to the G protein, okay, as soon as it comes and binds to G protein, what will happen? This G alpha क्या होगा? This alpha जो होता है alpha subunit, it has got its own GTPase activity. So what will happen? Okay, so this G alpha, this GDP will convert into GTP. तो binding के साथ साथ GDP will come into GTP. जैसे GTP convert हो जाएगा, G alpha will become active and it will separate from the G beta and G gamma. Fine. तो क्या हो रहा है यहाँ पे? यहाँ पे क्या हो रहा है कि जो GDP है, it's converting into GTP and then active हो जाता है. So G alpha 80 GTP is now in an active form. So this will have its own responses. You can see effectors out here, PKA, PYK, MEK5. Okay, so they will have it has its own responses, and G beta and G alpha will have its own again responses, cellular responses. Okay, तो ये होता है. Once this is done, what will happen again? This G alpha, मैंने अभी कहा था G alpha has its own GTPase activity because of which they can again hydrolyze GTP to form again GTP. So once again the GTP is formed back to GTP after the, देखिए सब कुछ downstream process हो जाने के बाद, when the work is over, when GTP will again convert back into GTP. As soon as that happens, क्या होगा? G alpha will again go and bind with the beta and gamma subunits, and again it will form the inactive G protein trimer. So this is the G protein activation. Okay, and also I've told you about the termination process also. Now let's come to the steps. I already told you. First step is ligand binding. Second is G protein activation, already explained. Now third is the effector activation. So what happens out here is that once the alpha subunit is activated, I told you they will interact with the G, they will interact and they will have their own process occurring out here. So क्या होगा? Then this G protein active form, it will go and bind to the adenylyl cyclase. यहाँ पे हम लोग बात करें about a certain pathway. Okay, तो इस pathway में क्या हो रहा है? There, see, after the G protein is activated, they go and bind to the receptors. मैंने आपको पहले बताया है कि they will go and bind to some of the effectors, not receptors, effectors. So out here, some of the common effectors are adenylyl cyclase, then we have phospholipase C, and we also have this ion channels. Okay, so once this effectors will go and bind with this activated G protein, it will lead to the generation of the second messengers like CAMP, IP3, DAG, which will again lead to formation of different type of like they will again form different things. So out here CAMP is forming protein kinase A. Okay, so यहाँ पे हो गया. Then protein kinase A is again forming phosphorylated protein again, which is giving a cell response. So this is the process that is occurring out here. एक बार ये सब हो जाता है and then again it leads to termination of the signal. तो मैंने already आपको बताया है क्या होता है. GTPase activity is there, GTP is converted to GDP, and then again they go and bind back. So this is the process that is occurring in the GPCR, or you can say G protein coupled receptor signaling pathway. Okay, तो याद रखिएगा CAMP अगर हुआ, it will activate the protein kinase A. अगर आपका IP3 हुआ, then IP3 क्या होगा? It will lead to release of calcium ions. अगर DAG हुआ तो क्या करेंगे वो लोग? प्रोटीन काइनेस C, PKC को वो लोग एक्टिवेट करेंगे. Okay, what are the other signaling pathways? Receptor tyrosine kinase pathway. I've already showed the diagram out here. See the first diagram, you can see in the first diagram out here, this is the receptor tyrosine kinase. So यहाँ पे क्या हो रहा है कि tyrosine kinase activity को activate हो रहा है. Okay, so this receptors they will again phosphorylate each other. I already told you कि जो यहाँ पे जो केस है RTK के केस में क्या होता है, they phosphorylate each other and also the other proteins. इसके बाद क्या होता है कि there are some downstream signaling events. Okay, example is RAS-MAPK pathway. I'll explain that to you in my future slide. Okay, so some examples I've given out here about this RTK receptors. Okay, signaling pathway के जो receptors हैं वो वाले. Then also I mentioned about ligand gated ion channels. I've already explained to you about it. Intercellular receptors मैंने already आपको explain कर दिया है. They are normally, these receptors are present in the cell, or you can say cytoplasm or nucleus. तो क्या होगा? Ligands bind होगा, they will go into the inside the cell membrane. Okay, और क्या होगा? Nucleus के अंदर में they will form this transcription with the help of some transcription factors. Okay, so ये होता है आपका intracellular receptors. Now coming to the calcium signaling pathway, I told you I'll explain to you about some pathways. So in this one what happens is that calcium cell के बाहर में present होता है. From outside this calcium will enter the plasma membrane. Okay, then what happens? This G protein coupled receptors, they will activate the phospholipase C beta, PLC beta, and RTK. Okay, and RTK will activate the PLC. Already explained to you कि RTK क्या करेगा? PLC को activate करेगा. GPRCR will activate the for PLC. Okay, PLC beta, and RTK will activate the PLC gamma. See out here. First ligand binding होगा. Okay, number 2 is then the activation of RTK and also activation of this GPR. Okay, GPCR. उसके बाद क्या होगा? GPCR will activate the PLC beta and RTK will activate the PLC gamma. Okay, this will lead to cleaving of, or you can say breakdown of PIP2 into IP3 and DAG. Already आपको मैंने बताया है कि जब ये होता है तो क्या होगा कि Ca2+ will be released. Okay, नंबर 4 से आप 5 में आ सकते हैं। देखिए नंबर 5 में क्या हो रहा है, दिस विल रिलीज Ca2 प्लस आयंस, फाइन। और उसके बाद क्या होगा कि जो IP3 जो रिलीज हुआ था, ये आपको क्या किया, Ca2 प्लस किया है, ओके। और DAG क्या करेगा, PKC, मैंने ऑलरेडी बताया है कि DAG विल एक्टिवेट प्रोटीन काइनेस C, ये हो गया। एंड देन ये जो आपका फॉर्मेशन हुआ है Ca2 प्लस सेंसिटिव DAG काइनेस, ये क्या करेंगे, DAG को फॉस्फोराइलेट करेंगे, फॉस्फोराइलेट करके इट विल लीड टू फॉर्मेशन ऑफ फॉस्फोटाइडिक एसिड एंड DAG लाइपेज, रिमेंबर DAG काइनेस, ओके, DAG काइनेस फॉस्फोराइलेट करेगा DAG टू फॉस्फोटाइडिक एसिड, और DAG लाइपेज विल कन्वर्ट DAG टू एरोकिडोनिक एसिड, ओके। इसके बाद क्या होगा, देयर विल बी सम फॉर्मेशन ऑफ बायोएक्टिव मॉलिक्यूल्स, ये चीज याद रखिएगा, ओके। सो आई हैव ऑलरेडी टोल्ड यू अबाउट Ca2 प्लस रिलीज, ओके ना। यहाँ पे एक और चीज है कि Ca2 प्लस सेंसिटिव प्रोटीन फॉस्फेटेज, इसका नाम है कैल्सिन्यूरिन, CANB बोलते हैं। दिस विल एक्टिवेट द ट्रांसक्रिप्शन फैक्टर NF80 विच प्लेज अ वेरी क्रिटिकल रोल आउट हियर। एंड अगर कभी भी ये Ca2 प्लस सिग्नल्स, इफ देयर इज अ डिसरेगुलेशन ऑकरिंग, दिस may lead to cardiac disease, schizophrenia, bipolar disorder and Alzheimer's disease. So you see, calcium signaling pathway is very important for the cell, okay. Now moving on to the RAS-MAPK pathway. So this RAS-MAPK pathway is also known as RAS-RAF-MEK-ERK pathway. Okay, तो ये किस में हेल्प होता है, सेल ग्रोथ में, प्रोलिफरेशन, डिफरेंशिएशन एंड सर्वाइवल में। सो अगर इसमें कुछ प्रॉब्लम होता है क्या होगा, कैंसर डेवलपमेंट, वेरी इंपॉर्टेंट, ओके, क्योंकि जो रास है ना, ये रास बहुत इंपॉर्टेंट चीज है। अगर कोई भी म्यूटेशन हुआ इसका ड्यूरिंग इन सेल साइकिल, इट विल लीड टू कैंसर डेवलपमेंट, ओके। नाउ व्हाट आर द स्टेप्स आउट हियर फॉर RAS-MAPK पाथवे, लेट्स सी। नाउ ऑलरेडी पहले मैंने आपको सारे स्टेप्स ऑलमोस्ट सेम है। पहले आपका क्या होगा, पहले लिगैंड बाइंडिंग होगा, ओके। उसके बाद क्या होगा, एक्टिवेशन होगा रिसेप्टर का। इन दिस केस सिंस इट्स अ RAS-MAPK, यहाँ पे क्या होता है, टायरोसिन काइनेज, रिमेंबर, ओके। टायरोसिन काइनेज एक्टिवेशन यहाँ पे होगा, ओके। टायरोसिन काइनेज एक्टिवेशन यहाँ पे होगा एंड इट विल then lead to the activation of again RAS, fine. So यहाँ पे receptor dimerization होगा, autophosphorylation होगा and this will lead to RAS activation. RAS activation के बाद क्या होगा, आपका RAF activation होगा, okay. So remember कि जो आपका RAS, ओके, यहाँ पे देख सकते हैं आप, RAS से जब RAF होता है तो क्या होता है कि there is a conversion of GDP to GTP, okay. तो जब ये होगा तो ये एक्टिवेट हो जाएगा, एक्टिवेट होके क्या होगा, राफ होगा, राफ फॉर्मेशन होगा, राफ को एक्टिवेट कर देंगे। तो एक बार रास एक्टिवेट हो गया, फिर रास के बाद राफ एक्टिवेट होगा, आफ्टर राफ अगेन MEK एक्टिवेट होगा, यू कैन सी द प्रोसेस आउट हियर, ओके। नाउ MEK क्या है, माइटोजन एक्टिवेटेड प्रोटीन काइनेस, ओके। तो आफ्टर ये MEK एक्टिवेशन हो गया, इट विल लीड टू ERK एक्टिवेशन, ओके। देन अगेन ERK के बाद क्या होगा, आपका सेल्युलर रिस्पॉन्सेज, और यू कैन से डाउनस्ट्रीम प्रोसेसेस। So this is a pathway which is occurring. This will lead to many type of regulation of cell, like your cell proliferation, differentiation, and survival. Now we come to JAK-STAT pathway, JAK-STAT pathway. तो यहाँ पे JAK क्या है, Janus kinesis, और STAT क्या है, signal transducer and activator of transcription. तो ये भी क्या है, यहाँ पे भी आपका बहुत सारा प्रोटीन इंटरेक्शंस होता है, जो क्या होता है बाहर में, okay, outside the cell if there are some chemical signals that are being transmitted to the nucleus of the cell, okay. तो यहाँ पे भी it will help in the immunity, cell division and metabolism regulation, fine. Now when the again same like before, ligand has to bind, the first step is always the ligand binding. So one ligand will bind, it will activate this JAK receptor C, it will activate this JAK receptor C. Receptor is already present here. This receptor is like attached with this JAK, okay. They are together associated with JAK. So when ligand will bind here, this receptor JAK will get activated, and when they will activate, they will phosphorylate the receptors. This JAK will phosphorylate receptors, and it will lead to this STAT phosphorylation. एक बार STAT phosphorylation हो गया, then they will dimerize. Okay, you can say dimerize out here, and then this dimerized STAT proteins, they will go to the nucleus, and then they will help in the gene transcription process. Okay, so this example can be seen in cytokine signaling and in interferon signaling. ये क्या कहाँ पे होता है, ये immunity में होता है ना, interferons and all cytokines, okay. Now कमिंग टू द टू कंपोनेंट सिस्टम. टू कंपोनेंट सिस्टम क्या होता है कि देखिए, the bacteria and plant, when they respond to the external environment, when they respond to the environment changes, then they do it with the help of this two component systems. यहाँ पे क्या है बेसिकली, यहाँ पे दो component होते हैं. एक component है आपका sensory kinase, so you can say sensor, which is the transmitter domain, and then there is also the response regulator, जिसको receiver domain भी कहते हैं. और यहाँ पे क्या होता है बेसिकली, जो सेंसरी काइनेस है, जो बाहर में जो स्टिमुली है, जो बाहर में जो when there's a change in the environment, it will detect that change, it will detect that stimuli. Okay, it will pass on to the transducer inside, it will pass on to the transmitter domain inside, which is the histidine out here. Okay, then what will happen, this histidine, then histidine, it will pass on to the response regulator, which is out at the aspartate. Okay, so from the histidine part, it will move on to the receiver part, that is the aspartate out here, which is also known as the response regulator, and this is how this is the expression of the bacterial genes. Okay, so this is all about your two component system. Classes will be in bilingual number 2. Okay, so this is all about a two component system. Fine. Now if you see out here in bacteria and fungi out here, see there are two type. Okay, simple and hybrid. Hybrid में कुछ नहीं है, there's just a repeating. Okay, in hybrid disease you can see there's additional round of phosphodilay, and in simple there is no additional rounds occurring. Okay, so this additional rounds, this is occurring in the eukaryotes, and this is occurring in the prokaryotes. Fine. So in bacteria, you can see the right hand side diagram out here. In bacteria यहाँ पे क्या हो रहा है, this two component system, it's leading to some process known as the downstream process. यहाँ पे क्या क्या हो रहा है, it's helping in the antibiotic resistance, chemotaxis, and virulence. Okay, this two component system is helping the bacteria to in this processes, you can say the responses. Okay, and in fungi, or you can say in eukaryotes plants, they are helping in different type of functions such as the transcription, also the drug resistance, virulence, adhesion, and morphogenesis. So this two component system is helping in this type of responses. Okay, now coming to the bacterial chemotaxis and quorumsensing, this is the last part. So we know that these two process, okay, chemotaxis and quorumsensing, these are two ways. ये दो तरीका है जिसके वजह से, जिसके जो use करके bacteria, they communicate and they move in the environment. See, bacteria has to communicate with the external environment. They have to move in their environment. So they use these two methods, bacterial chemotaxis and quorumsensing, to do that. Okay. So what is bacterial chemotaxis? See, chemo means what? Your chemical. So chemotaxis means out here when bacteria can move either towards a chemical gradient or it can move away from the chemical gradient. तो अगर एक chemical signal सामने होता है तो bacteria either उसके पास जा सकता है towards, और वो उससे दूर भी जा सकता है, repel भी हो सकता है. Okay, so ये जो है this is known as chemotaxis, ये ability of the bacteria. तो ये आपका maximum example है, जैसे कि gut bacteria, gut में जो होता है. Okay, तो ये कैसे बैक्टीरिया करते हैं, विद द हेल्प ऑफ टू मोड्स ऑफ स्विमिंग, रन एंड टम्बल. वो जाते रहते हैं, स्टिमुली को देखते हैं, अपना या तो अपना मोड चेंज कर देते हैं, दे चेंज द डायरेक्शन और दे विल मूव इन डिफरेंट डायरेक्शन. ओके, सो आइदर दे विल बी अट्रैक्टेड और दे विल रिपेल. ओके, देन कमिंग टू कोरम सेंसिंग. कोरम सेंसिंग क्या है, दिस इज आल्सो एन एबिलिटी ऑफ अ बैक्टीरिया to communicate with each other. Bacteria, they will communicate with each other and they will produce, okay, either they will produce some chemicals or they will send some small molecules, and this is how they will communicate with each other, with the bacteria. Okay, इससे क्या होता है, they will help the bacteria. Okay, this process will help the bacteria coordinate and they will help in the gene regulation process. Okay, तो क्या होता है, in biofilm formation, the secretion of virulence factors. ठीक है, तो ये सब चीजों में help करता है. They will also help the individual bacteria जो कि colonies में present होते हैं to coordinate among each other. Okay, and form some process known as bioluminescence, जो कि देखा जाता है विब्रियो कॉलरे में देखा जाता है. Okay, so this bioluminescence, then you have virulence, then conjugation, competence, biofilm formation already बोला है. So these all type of functions can be done with the help of this कोरम सेंसिंग. तो मैंने यहाँ पे डायग्राम में दिया है कि कैसे होता है कि बैक्टीरिया का क्या करते हैं, दे विल सेंड सम मॉलिक्यूल्स, जैसे कि यहाँ पे ऑटो इंड्यूसर दिख रहा है. ओके, सो ऑटो इंड्यूसर हो रहा है and then what happens, they will activate the behavioral genes. Okay, they will, okay, now receive करेंगे उसको bacteria and they will get activated, and then what will do, they will help in the communication process. Okay, So this is all about the signal transduction pathway. So here I've put some questions for you to practice. So let's discuss it. Okay. So first I've told you which of the following is not a which of the following is not a common secondary messenger. So यहाँ पे कौन सा common secondary messenger नहीं है? CAMP, CGMP, IP3, ATP. कौन सा होगा? ATP obviously, right? Then we have the which type of receptor directly regulates the opening of an ion channel upon ligand binding. कौन सा यहाँ पे होगा? G protein है, receptor tyrosine kinase है, ligand gated है. तो ion channel में कौन सा होगा? Ligand gated ion channel. Number C. Okay, which of the following signaling molecules acts as a neurotransmitter and a hormone? कौन सा यहाँ पे होगा? आपको पता है ना? Epinephrine. Okay. Then which signaling pathway is primarily involved in the regulation of glycogen metabolism? कौन सा होगा यहाँ पे? MAPK, PI3K. कौन सा होगा यहाँ पे? आप बोल सकते हैं ग्लाइकोजन मेटाबॉलिज्म. ओके, तो ये आप खुद करेंगे. ठीक है. नाउ विच ऑफ द फॉलोइंग इज नॉट अ कैरेक्टरिस्टिक्स ऑफ रिसेप्टर टायरोसिन. ये मैंने आपको नहीं बताया ग्लाइकोजन मेटाबॉलिज्म, पर जो करता है इसमें वो क्या क्या करता है आपका? वो ग्लाइकोजन मेटाबॉलिज्म में हेल्प करता है, ग्लूकोसिनेसिस में भी हेल्प करता है. देयर आर मेनी थिंग्स दैट इट इज हेल्पिंग इन. ओके. सो विच ऑफ द फॉलोइंग इज नॉट अ कैरेक्टरिस्टिक्स ऑफ रिसेप्टर टायरोसिन काइनेज. यहाँ पे कौन सा रिसेप्टर नहीं है ये भी आप बताएंगे. फाइन. ये आंसर भी मैं आपको नहीं बताऊंगी. दो मैंने आपको 3 बताया है, 2 आपके लिए छोड़ा, आप तो खुद सॉल्व करेंगे. नेक्स्ट, विच ऑफ द फॉलोइंग सेकेंडरी मैसेंजर इज प्रोड्यूस्ड बाय एंजाइम फॉस्फोलिपिड सी. ये भी मैंने आपको ऑलरेडी बताया है इन माय क्लास. ये भी आप खुद करेंगे. विच ऑफ द फॉलोइंग सिग्नलिंग मॉलिक्यूल्स कैन डिफ्यूज टू द प्लाज्मा मेम्ब्रेन एंड बाइंड टू इंटरसेल्युलर रिसेप्टर्स. ये आंसर मैं आपको बता देती हूँ. द आंसर इज आउट हियर, नाइट्रिक ऑक्साइड. ओके, सी. नाउ which of the following is a key protein involved in the regulation of the cell cycle and is often mutated in cancer. ये मैंने आंसर मैंने ऑलरेडी आपको बताया है while teaching about a certain pathway. हिंट दे दिया मैंने आपको. You have to tell the answer. Fine. Which of the following signaling pathways is involved in inflammation? ये भी आपको खुद बताना पड़ेगा. चलिए मैं आपको बता देती हूँ इसका आंसर. इसका आंसर है ANF-KB pathway. Okay. Now next, which of the following is not a common mechanism for. अच्छा, इसका तो आंसर मैंने यहाँ पे नीचे दे ही दिया आपको. चलिए कोई बात नहीं. ओके, सो विच इज नॉट अ कॉमन मैकेनिज्म फॉर टर्मिनेटिंग अ सिग्नल ट्रांजिशन पाथवे. मैंने आपको ऑलरेडी बताया है कि टर्मिनेशन का प्रोसेस क्या है. तो एक जो है प्रोसेस ये मैंने आपको नहीं पढ़ाया है, इसलिए मैंने यहाँ पे आंसर दिया आपको. इंक्रीज्ड सिंथेसिस ऑफ सिग्नलिंग मॉलिक्यूल्स. कभी कभी क्या होता है ना कि टर्मिनेट करने के लिए सिग्नलिंग मॉलिक्यूल्स जो है वो बहुत ज्यादा इंक्रीज अमाउंट में वो सिंथेसिस होते रहते हैं. तो क्या होता है कि ये जो प्रोसेस है ना ये कॉमन नहीं है, क्योंकि अभी बाकी जो आप यहाँ पे देखेंगे A, B and C वो कॉमन है, पर D जो इतना कॉमन नहीं है. तो इसलिए क्योंकि मैंने आपको बताया नहीं इसके बारे में, since I have not told you about this, so I have given you the answer. Okay, so all the best for your CSI exam and do solve the questions and do comment me the answers. Okay, if you like the class, then do click on the like icon, share it with your friends and do subscribe to my channel. Okay, so thank you so much for attending today's watching today's video, attending today's class. So thank you so much. Have a very nice day. Bye bye.